Scientists have found that inactivated and mRNA vaccines trigger different T-cell responses in fighting serious COVID-19 virus. Inactivated vaccines elicited a broader immune response against different proteins on the virus, while mRNA vaccines can only induce T-cells targeting the virus's spike protein.
And for what that all means for the future of vaccine strategies, we're joined by Dr. Anthony Tanoto Tan, Senior Research Fellow from the Emerging Infectious Diseases Programme at Duke-NUS Medical School. Dr. Tan, first of all, take us back to a discussion that we had right at the beginning of the pandemic on what T cells are and how they help our bodies fight off viruses.
Yes, so the vaccine-induced response has two arms. One is the one that most people are more familiar with, which are the antibodies. The antibodies bind to the viruses and they stop the virus from infecting a cell. So the other arm, let's call it the less known cousin, are the T cells. The T cells do not neutralize the virus. They do not detect the virus. What they do is they clear the cells that have been infected with the virus. So the real difference between the two of them is that the antibodies prevent infection, while the T cells prevent someone from falling severely ill.
Dr. Tan, can you explain to us why you chose to test for the differences in T cell responses and what's causing the different T cell response in different vaccines?
So why we chose to test the T cell responses. So as you know, with the emergence of the Omicron variant or the Omicron lineages of viruses, these viruses are getting better at escaping the antibody response. So clearly, facing with a virus like that, we are paying more attention to not preventing infection, but more on ameliorating the severe disease. So really, these are associated with T cells, and we start to look at the T cell response from that perspective. As to why they are different, basically, it might get a little bit complicated, but basically what it means is that the vaccines stimulate different T cells in the body differently. So depending on the vaccine technology, you can stimulate different subsets of T cells.
All right, so I'm with you so far, but now I've got to bring in another word, another term, inactivated vaccines. We talked earlier about them eliciting a broader immune response. Does that then mean that they are better foot soldiers in the prevention fight?
So this is a tough question to say because the broader aspect or the broader immune response induced by inactivated vaccines is not just the only difference between mRNA and inactivated vaccines. There are several other differences. And when we say you want a better protection, it's actually a net effect of all these variables. So it's difficult to predict which vaccine is better and which vaccine is worse. Instead, I will draw the argument or the discussion towards instead of pitting these different vaccines head to head and trying to see which one's better, why don't we consider all these vaccines as assets that we have? And we know what are the pros and cons of this vaccine, what are the advantages and disadvantages, and we try and utilize each of the vaccine's advantage and perhaps use them in combination in this long-drawn fight against SARS-CoV-2.
Dr. Tan, does that strategy still hold validity even as a virus is mutating? And I mean, with regards to the COVID-19 virus, it appears to have mutated quite rapidly over the past couple of years. I mean, this has been the experience that we've had. Is that argument still valid?
In a way, yes, because the biology of T cells itself, the way it recognizes and the way it gets activated is more resilient to mutations than compared to the biology of how antibodies recognize the virus itself. So even when the virus starts to mutate, when we start to have the alpha, beta, so on until Omicron and now XBB, we have seen that the T cell response can remain largely preserved against these different variants. In contrast, antibodies, or at least some of the antibodies, do exhibit a complete inability to recognize some of these variants itself.
Earlier, Doctor, you spoke about not having to choose one or the other, but to use all of them as a combination, as a suite of these weapons we have against the pandemic. Now, most of us are already vaccinated with either this or that type of vaccine. So how then do you combine them in a way that the T-cell immune responses of both the inactivated vaccines and the mRNA vaccines work together for future pandemics?
So perhaps I'll give an example. So like, for example, most of us have already received a primary vaccination with the mRNA vaccines. But we know that the mRNA vaccines are targeting only the spike protein itself. So perhaps to give a broader protection, one could receive the inactivated vaccine, which we now have shown that it could induce a broader T-cell response targeting different proteins of the virus. So in that end, you can now have an individual that is initially protected against spike with a highly efficient mRNA vaccine, and then now you receive the inactivated vaccine that broadens the T-cell response against SARS-CoV-2. So this is just an example of how we can utilize the pros and cons of each of the vaccine to try and combat the SARS-CoV-2 pandemic.
Some very interesting and useful applications for this. Dr. Tan, thank you very much for joining us to shed light on this. That was Dr. Anthony Tanoto Tan, Senior Research Fellow from the Emerging Infectious Diseases Programme at Duke-NUS Medical School.